GCSE Biology, higher. Question 1. Plants transport water and mineral ions from the roots to the leaves. What process do plants use to take minerals into the root? Active transport. Where you see a tick in the answers, that's a point. Describe how water moves from the roots to the leaves. By transpiration stream, 1 mark. Through the xylem, 1 mark. Plants lose water through the stomata in their leaves. The epidermis can be peeled from a leaf. The stomata can then be seen. Describe how the student could have collected the data. So we've got 3 marks available. You can see there's a number of different points you could have made. First of all, place the epidermis onto a slide. Count the number of stomata in that area. Very, very simple. You could repeat the experiment in 4 more areas and take a mean. You've got to repeat the experiment on the other side of the leaf in order to make your comparisons. What is the median number of stomata on the upper surface of the leaf? The median value, as you know, is the middle value, that is 1. Calculate the value x in the table. Looking here, x is the mean of all of these values, so we simply add up all the values together and divide it by 5. You can't have a fraction of a stomata, so you've got to make sure you think about what you're actually doing. So 1 mark for working, 1 mark for answer, 2 marks in total. The plant used in this investigation has very few stomata on the upper surface of the leaf. Explain why this is an advantage. Less stomata means less water was lost. This means the plant will not wilt. 2 marks there. Okay, not just for those words, for those sentences. Okay. Question number 2. The tobacco mosaic virus, TMV, is a disease affecting plants. All tools should be washed in disinfectant after using them on plants affected with TMV. Suggest why? To kill the virus or prevent it from spreading. The slash means or. 2.2. Scientists produced a single plant that contained a TMV-resistant gene. Suggest how the scientists can use this plant to produce many plants with the TMV-resistant gene. So, this is basically, girls, tissue culture. Take a section from the stem, you could have said meristem. Perform a tissue culture. That's where you put it out into lots of different petri dishes. Or you could have said perform a cutting. Yeah, you're basically just talking about asexual means of reproduction here. Question 2.3. Some plants produce fruits which contain glucose. Describe how you would test for the presence of glucose in a fruit. With the glucose test, you need to heat the mixture, add some Benedict solution, and it will go from blue to orange if there is sugar present. Okay, so most of the time, girls, you've just got to remember to say the actual result. Lots of people forget to do that. 2.4. TMV can cause plants to produce less chlorophyll. Explain why plants with TMV have stunted growth. So, there's less photosynthesis due to a lack of chlorophyll. It's told you that up there. So less glucose is made. So less energy is released for growth. or less amino acids are made for growth. Amino acids are made into, of course, proteins. Proteins are what are needed for the plant to grow. Glucose is needed to make cellulose or proteins. So any of those in any order, really. Question 3. Microorganisms cause infections. The human body has many ways of defending itself against microbes. Describe 2 ways the body prevents the entry of microorganisms. Okay, so this isn't about killing them when they're in the blood. This is about preventing their entry. So we've got number 1, acid in the stomach killing pathogens. Number 2, skin forming a barrier to the pathogens. Hairs on the nose or the trachea trap the particles, or if you'd said cilia there, that would have been fine. Mucus is produced to trap pathogens. Cilia, okay. You could also have talked about scabs here, preventing the entry of pathogens. So, okay, in 2014, the Ebola virus killed almost 8,000 people in Africa. Drug companies have developed a new drug to treat Ebola. Explain what testing must be done before this new drug can be used to treat people. Okay, so we've got our preclinical trials. They always come as our first bit. That's testing drugs on cells or tissues or live animals. Okay, this is testing toxicity. It's not testing side effects at the moment. Or it could be testing for dosage, the correct dosage, or efficacy, which basically means do they actually work? Testing the drugs to see if they're all right. After that, as you know, we move into our clinical trials. Okay, and those clinical trials are testing the drugs on healthy volunteers, first of all, okay, at very low doses, so that you can monitor those side effects. Then the trials are done on patients to find the optimum dosage and to find the efficacy, the optimum efficacy. Okay, so you've got 2 levels there, healthy volunteers, then patients. It's worth at this point discussing the fact that a double-blind trial would have been done. A placebo would have been used that does not contain the actual drug. And this should be random so that you can test to see the effect of the drug and prevent any false claims. So let's have a look at point allocation. Mention of a preclinical trial. What is that preclinical trial? Why are you doing that? Okay, so any of those in any order. Mention of clinical trials. Okay, coming after, next. Testing them on healthy volunteers first, at low doses, so that you can monitor side effects. Trials are then done on patients to find the optimum dosage or the efficacy. And any mention of a double-blind trial in order to prevent false claims. Okay, some kind of explanation of what that is would be good. It's hard to mark this kind of question, but just have a look in this case for the ticks, please. Question number 4. All living cells respire. Respiration transfers energy from glucose for muscle contraction. Describe how glucose from the small intestine is moved into a muscle cell. So, first of all, the glucose moves into the blood by diffusion. Okay? Then, once it gets to the cell, the blood delivers glucose to a muscle in the capillaries, again, by diffusion. So it's just about talking about how it gets from the small intestine to a muscle cell. Okay? This is an investigation to look at how yeast cells respire. What is the purpose of the liquid paraffin? So the liquid paraffin is up here. The yeast are in here. You want them to respire anaerobically. So it's done to stop air getting in. That would be the only useful thing here. The rest of them, paraffin, you know, doesn't affect temperature. Water's not going to come from anywhere. Evaporation isn't going to affect respiration anyway. So think about it logically. So, you've been told the indicator solution in tube B shows the changes in concentration of carbon dioxide. The indicator goes blue when carbon dioxide is low, green when it's, blue when it's very low, green when the concentration is low, and yellow when it's high. What colour would you expect the indicator in tube B to go during the maximum rate of respiration? Now you know respiration produces CO2. So therefore it must go yellow because that's going to be surrounding here. These bubbles are the gas produced by the yeast. Question 4.4. Suggest how the experiment could be changed to give a reproducible way to measure the rate of reaction. So here they're just looking using an indicator. If you want to know anything about volumes, you're going to have to collect the CO2 in a measuring cylinder, which we have done before, and measure. If you're looking at rate, it's a volume in a particular time. Okay, so I would expect you to talk about collection and volumes and then how to actually conduct or calculate a rate from that. So compare anaerobic respiration in yeast with anaerobic respiration in a muscle cell. Now if I were you, I'd make sure, if you haven't got this down already in your notes, you need to make sure you go back and look at it, okay? So there's a difference. So we've got yeast, which is the breakdown of glucose. If you have a look at what I'm writing down here, breakdown of glucose, this is respiration, into ethanol and CO2. In a muscle cell, which is a normal animal cell, and any animal cells, we've got the breakdown of glucose, you know, into simply lactic acid. Remember, we're not looking at aerobic respiration. Both of these are going to release energy because respiration releases energy. Okay, but there is a subtle difference between the two. So if we're comparing them, yeast produces ethanol, there's my comparative word, but muscles produce lactic acid. Yeast produces CO2, but muscles do not. There's my comparative word. You could have used whereas as well. If we're comparing them, you don't have to just do differences. Okay, it's anaerobic respiration, so we're comparing them. We're saying that both of them release a small amount of energy. That's the similarity there. Okay.